Today we are going to talk about lipids. Lipids play many important roles in the human biology and in the human health. For example, cholesterols of different types may either contribute to the development of heart disease or may prevent it. So lipids are biological molecules that contain either fatty acids or a steroid nucleus. Steroid nucleus is shown here. Lipids can be soluble in organic solvents, however they are not soluble in water. Their name is derived from the Greek word lipos, which is a Greek word for fat. Lipids play crucial roles in the formation of cell membranes. They can be fat-soluble vitamins, and they are essential for the production of steroid hormones. Lipids can be divided into two large groups based on the chemical structure. Waxes, fats, oils, and phospholipids are all esters. These esters can be hydrolyzed, and that hydrolysis will produce fatty acids and some other molecules. Steroids that are also lipids do not contain any fatty acid chains, and they cannot be hydrolyzed. The main characteristic is that steroids consist of steroid nucleus of 4 fused carbon rings with some side chains attached. So this flowchart shows you different types of lipids, fatty acids that are essential for the formation of waxes, triacylglycerols, glycerophospholipids and sphingolipids, and steroids. You can see the commonalities in structure between these 4 types, and steroids are vastly different. First, we're going to talk about fatty acids. Fatty acids can be saturated or unsaturated, and one of the key properties of saturated fatty acids is that molecules of saturated fatty acids have fairly regular shapes, and due to these regular shapes, these molecules stick closely together like chips in a can. Unsaturated fatty acids have more irregular shapes, so they would look like chips that do not fit together closely like in the bag of chips. So fatty acids are long chains, unbranched, and that is important. They are unbranched chains of carbon atoms with a carboxylic acid group at one of the ends. Typically, these chains have 12 to 18 carbons, and because of these long carbon chains, fatty acids are insoluble in water. Fatty acids are considered to be saturated when there are no double carbon-carbon bonds in the carbon chain, and they are considered to be unsaturated when these carbon chains contain one or more double bonds between 2 carbons. So when you look at the formula of a fatty acid, you can draw it like all the way. Sometimes you can shorten it by taking methylene groups and just giving a number of that, or you can draw a skeletal formula where you do not put any carbons, but you presume that every end and an angle of the line is a carbon atom. So as we mentioned before, fatty acids can be saturated. Examples are shown in this table with different number of carbons, or they can be unsaturated. Unsaturated fatty acids, if they contain only one double bond between carbons, are called monounsaturated, such as palmitoleic acid or oleic acid. You can see here that the first number in that designation shows you how many carbon atoms are in the molecule, and the second number shows you how many double bonds are there. Or they can be poorly unsaturated, when there are at least 2 or more double bonds between 2 carbons in the carbon chain. So you can see linoleic, linolenic, and arachidonic acid that contain multiple double bonds in the chain. Unsaturated fatty acids can form trans and cis isomers. These trans and cis isomers shown here, you can see that in cis isomer 2 hydrogens are located on the same side of the double bond, that's cis, and in trans oleic acid hydrogens are located on the opposite sides of the double bond. Practically all naturally occurring fatty acids that are unsaturated, they have cis double bonds. So we can synthesize some fatty acids from carbohydrates or other fatty acids, but there are certain poorly unsaturated fatty acids that we cannot synthesize in sufficient amounts. These are some examples. You don't need to memorize them. What you need to know is that the poorly unsaturated fatty acids that we cannot, some poorly unsaturated fatty acids we can't synthesize, these fatty acids are called essential fatty acids. Let's talk about properties of saturated and unsaturated fatty acids. Saturated fatty acids contain only single carbon-carbon bonds, and they fit close together like shown in this illustration, which means that these fatty tails interact by strong dispersion forces. The strong dispersion forces hold them together really well. That means that saturated fatty acids have high melting points, and they're usually solid at room temperature. On the other hand, in unsaturated fatty acids, these double bonds in the cis conformation result in the bending of carbon chains, which means the molecules have irregular shape, and because of that have fewer interaction between the molecules. And that reduced number of interactions in unsaturated fatty acids between the molecules of unsaturated fatty acids reduces the melting point for such molecules. Another special type of molecules that are referenced as fatty acids are prostaglandins. Prostaglandins are produced in cells. These are substances similar to hormones, also known as eicosanoids. These are formed from arachidonic acid. It is ultimately a source for prostaglandins. Arachidonic acid is polyunsaturated. It has 20 carbon atoms and 4 double bonds. And then 5-carbon ring that is a part of prostaglandins is usually modified by some substituents. They have these prostaglandins have multiple functions. They may change the blood pressure, they may stimulate contraction and relaxation of smooth muscles, they can even change the body temperature. So during the tissue injury, arachidonic acid, shown here, is converted to prostaglandins of various types, and they stimulate inflammation at the damaged area. Non-steroidal anti-inflammatory drugs, such as ibuprofen or aspirin, they block the production of prostaglandins, reducing the pain, the fever, and the inflammation. So these are some examples of non-steroidal anti-inflammatory drugs, and they inhibit essential enzymes in the cell that generate prostaglandins. So fatty acids significantly contribute to various, human conditions. So turns out that unsaturated fatty acids may be more beneficial to human health compared to saturated ones. Multiple vegetables contain omega-6 unsaturated fatty acid, meaning that the first double bond can be found in the carbon-6 if you count from the methyl and the carbon chain. Another type of useful unsaturated fatty acid is omega-3 acid. They're pretty famous. First double bond occurs at carbon-3 if you count from the methyl end, and these fatty acids can be found in cold water fish. So it turns out that omega-3 fatty acid lowers the ability of blood platelets to sticking together in the unprovoked manner, and thus they reduce the possibility of blood clot formation. Blood clot formation, unprovoked blood clot formation, is directly linked to conditions like heart attack or stroke, and turns out that these are intrinsically connected with cholesterol. So excessive cholesterol in the blood results in the plaque formation on the walls of the blood vessels that increases the blood pressure, and it also leads to remodeling of the blood vessels, increased inflammatory responses in the blood vessels, formation of more plaques, and narrower vessels are easier to block with the blood clots. So all in all, omega-3 fatty acids seem to have beneficial effects on the cardiovascular health. Now we're going to talk about waxes and triacylglycerols, and we're going to start with waxes. They can be found in many places in nature: fruit surface, leaf surface, plant stems, and they're also found in the honeycombs. So waxes are essentially esters of saturated fatty acid on one hand and long chain alcohol on another. Each wax can contain, obviously, each molecule can contain from 14 to 30 carbon atoms. That makes them incredibly hydrophobic. So these are some typical waxes: beeswax, carnauba wax, jojoba wax. They're used in various products, from candles and shoe polish to soaps and cosmetics. And now waxes are not that particularly important for human health directly. Triacylglycerols are. So fatty acids in humans are stored as triacylglycerols or triglycerides. Triglycerides are glycerol esters that form, ester bonds with fatty acids. Glycerol is alcohol with 3 hydroxyl groups. So when three hydroxyl groups of glycerol react with a carboxyl group of 3 fatty acids, triglycerides are formed. The name is formed by changing glycerol to glyceryl and then name the fatty acids as carboxylates. So, for example, you have 3 stearic acid molecules and you have glycerol. You change glycerol to glyceryl, and each stearic acid is changed to stearate. So glyceryl tristearate. Now, sometimes triacylglycerols can be mixed when you have 3 different fatty acids that form triacylglycerol. It is totally possible. The main function for triacylglycerols in animals and humans is the storage of energy. Okay, major form of energy storage in animals and humans, especially in hibernating animals. These animals eat large amount of food that are high in calories during the summer months, and when external temperature goes down, the animals, such as a bear, go into hibernation. The body temperature reduces dramatically, cellular activity is reduced, respiration and heart rate are reduced, and the stored fat in the animal is the only source of energy for such process during the hibernation. Okay, so that's your triacylglycerol right there. That its function. Now, you at some point, probably not here, but probably will have to draw the structural formula of triacylglycerol. So how do you do that? Glycerol tripalmitoate, tripalmitoleate. So what do you need to know? You need to figure out what the components. So glycerol means there's a glycerol molecule, used to be. Tripalmitoleate contains 3 palmitoleic acid residues. So this is your glycerol residue, and those are 3 residues of palmitoleic acid. So now we're going to compare fats and oils. So what is the difference between the two? Turns out fats are usually solid at room temperature because they contain mostly saturated fatty acids, and fats come from animal sources, mostly shown here: cheese, butter, meat. Oil is usually liquid at room temperature because it contains mostly unsaturated fatty acids, and it is usually obtained from a plant source. So different types of plants produce different types of oils, like olive and peanut oil contain mostly monounsaturated fatty acids. Corn, cotton seeds, flower seeds, and flower oil are polyunsaturated. And palm and coconut oils are solids because, well, it kind of contradicts, but it's sort of an exception. For our intents and purposes, oils are liquid at room temperature. Palm and coconut oil are solids because they consist mostly of saturated fatty acids. So in terms of melting points, you can see that saturated fatty acids have higher melting points than unsaturated fatty acids, since they packed together in a more tight fashion. So here you can see comparison of the content of saturated, shown in blue, versus unsaturated, shown in red and yellow, in different products. And you can see that in solid food products, saturated fats are the predominant ones. Saturated fatty acids are the predominant ones. While in the liquid food products, mono and polyunsaturated fatty acids dominate. So now we're going to talk about the chemical properties of triacylglycerols, and we're going to start with the hydrogenation. Turns out that many products such as margarines and shortenings, they are actually derived from vegetable oils via partial hydrogenation. So in reactions of hydrogenation, double bonds of unsaturated fatty acids interact with hydrogen molecules, and that results in a conversion of double bonds to single carbon-carbon bonds. This reaction happens usually in the presence of a nickel catalyst by bubbling hydrogen gas through the heated oil. So here you can see that double bonds in glyceryl trioleate are being replaced by single bonds, resulting in glyceryl tristearate, converting 3 unsaturated fatty acids into 3 saturated ones. So in commercial hydrogenation, it is stopped before all the double bonds in a vegetable oil become completely saturated. And depending on how far the hydrogenation goes, if it is very partial, it results in a soft, semi-solid, fat-like tab margarine. If you saturate it more, it will generate products with a higher melting point, such as stick margarine. So we mentioned that unsaturated fatty acids can be cis and trans. So turns out, and remember, it's the side of the groups relative to double bond. Turns out that during hydrogenation, double bonds are converted to carbon-carbon single bonds, but some of the cis double bonds are converted to stable trans double bonds. They are chemically more stable, and that changes the overall structure of the fatty acid. Turns out that trans fatty acids, despite being unsaturated, behave just like saturated fatty acids. Even worse than that, they significantly increase the levels of low-density cholesterol and lower the levels of high-density cholesterol, significantly increasing the risk for the development of cardiovascular disease. So here you can see how cis oleic acid is converted, hydrogenated. You have desired saturated product, which is stearic acid. It's not a problem, but the problem is isomerization of cis oleic acid into a trans oleic acid, and this one significantly increases the risk of cardiovascular disease. So there's a very, very small amount of trans fat that can be found in products like milk, eggs, and beef, but most deep-fried foods, baked goods that used vegetable shortening in them, chips and crackers, margarines, and vegetable shortening itself, they contain substantial amount of trans fatty acids, and these foods should be avoided at all cost. Hydrolysis of triacylglycerol, acid hydrolysis, or hydrolysis with a lipase, results in the production of the molecule of glycerol and 3 molecules of fatty acids. Now, saponification is the reaction in which fat is hydrolyzed using base, not acid. So triacylglycerols can be saponified by sodium hydroxide into glycerol and sodium salts of fatty acids that were components of triacylglycerol. This is the process in which soaps are formed. If you use sodium hydroxide, that will give you a solid soap. If you use potassium hydroxide, it will give you softer liquid soap. So here you can see saponification reaction. You have glycerol and salts of fatty acids. This slide summarizes main reactions: esterification that results in the formation of triacylglycerols, hydrogenation that converts unsaturated fat to saturated fat, and hydrolysis and saponification of triacylglycerols. Now we're going to talk about phospholipids. Phospholipids is a family of lipids that are similar in structure to triacylglycerols. 2 main types of phospholipids are glycerophospholipids and sphingomyelin. Glycerophospholipids contain 2 fatty acid residues that form ester bonds with 2 hydroxyl groups, first and second, of the glycerol molecule. Third hydroxyl group in glycerol forms an ester with phosphoric acid, which forms another phosphoester bond with a molecule of amino alcohol. Sphingomyelin contains the molecule of sphingosine instead of glycerol. It contains one molecule of fatty acid, and it is also linked to phosphate, which in turn is linked to amino alcohol. There are 3 amino alcohols that are found in glycerophospholipids: choline, serine, and ethanol amine. Now we're going to talk about a couple of examples of glycerophospholipids: lecithin and cephalin. Lecithin on the left contains 2 nonpolar fatty acid chains, glycerol, phosphate, and highly polar choline. Cephalin contains 2 fatty acid chains, glycerol, phosphate, and highly polar ethanol amine. So glycerophospholipids have 2 key regions, polar and nonpolar regions, that allow them to interact with both polar and nonpolar substances. The polar head of glycerophospholipid contains the ionized amino alcohol and the phosphate portion. They strongly attracted to water. Hydrocarbon tail, well, consists of glycerol and 2 fatty acids. And fatty acids, as you remember, are soluble only in nonpolar substances, such as lipids, so they're nonpolar or hydrophobic. These lipids are the most abundant lipids in the cell membranes, and they play a critical role in the regulation of cellular membrane permeability. So these are some examples of glycerophospholipid. So this is the full structural formula, but if you look at these formulas, you will see the highlighted polar head and nonpolar tail. And sometimes they are pictured this way. So turns out that snake venom contains enzymes called phospholipases, which break down phospholipids in red blood cells and result in the hemolysis and symptoms of snake venom toxicity. Now let's talk about sphingomyelin. So sphingomyelin, the amine group of sphingosine, forms the amide bond with a fatty acid. You already know what amides are. And the hydroxyl group of sphingomyelin forms an ester bond with a phosphate. Phosphate in turn forms another phosphoester bond with either choline or ethanol amine. Sphingomyelins can be found in the myelin sheath around nerves and neurons, and it's critical for the function of the myelin in terms of protection of nerve cells and electrical insulation and increase of the speed of the nerve impulse. So this is a typical structure of sphingomyelin. You can see the sphingomyelin molecule here. Sorry, like this. That's sphingomyelin. This is the fatty acid, phosphate, and the choline residue. So now we're going to talk about steroids. So steroids are essential for production of various hormones, as well as for transport of cholesterol between the tissues and the liver. Steroid nucleus consists of 3 cyclohexane ring and 1 cyclopentane ring attached to each other, fused together. Rings are designated as A, B, C, and D. And carbon atoms, the numbering starts in ring A. There are 2 methyl groups in position 18 and 19. So here and here. And this is the formula of cholesterol. It is the most abundant, sorry, not here and here, but here and here. That's methyl groups. Cholesterol is the most important and abundant steroid. It has a hydroxyl group, which makes this part polar. It's double bond between carbons 5 and 6. It has methyl groups at carbons 10 and 13, and it has a long alkyl chain at carbon 70. Cholesterol can be obtained from milk, meat, or eggs, so basically animal products. It is synthesized in the liver. As a matter of fact, 85% of human cholesterol is synthesized in the liver. It is essential for cell membranes, for the proper function of brain and nerve tissue, for synthesis of steroid hormones, and for synthesis of vitamin D. When it reaches high levels in the blood, it clogs arteries, forming plaques. You can see an open artery, and you can see an artery with a cholesterol plaque pretty much fully obstructed by cholesterol. It's considered elevated when the plasma cholesterol is above 200 mg per deciliter. However, more and more evidence emerges suggesting that total levels of cholesterol are kind of irrelevant. It is mostly synthesized in liver, partially obtained from foods. So this statement is wrong. A diet that is low in foods containing cholesterol and saturated fats, it does not affect serum cholesterol level and does not play any role in the cardiovascular disease. It's cholesterol content. We're going to skip it, and we're going to talk about bile salts. So bile salts are synthesized in the liver and stored in the gallbladder. They have polar region and non-polar region, and that allows them to make fat soluble in water, which helps in the fat digestion. Okay, the ability to solubilize fat. They also help in the absorption of cholesterol. Excessive cholesterol in the bile results in the formation of gallstones. This is the structure of a typical bile salt, sodium glycocholate. This is from glycine. This is from cholic acid, a bile acid, and that's a bile salt. Basically, a combination of an amino acid and a bile acid. So we're going to say a few words about lipoproteins. Lipids that are required as an energy source, they're nonpolar. And to transport them in the body, you have to make them soluble. You can make them soluble but by combining lipids with glycerophospholipids and proteins, which forms water-soluble complexes that are called lipoproteins. So lipoproteins, their function is to transport fats, nonpolar lipids in the body, transport them to cells. Lipoproteins themselves are soluble because of their unique structure. So it turns out that inside of the lipoprotein, you can find molecules of cholesterol esters and triacylglycerols. These molecules are nonpolar, but on the surface of the lipoprotein, you can find more polar molecules like phospholipids, cholesterol, with OH groups. So surface is polar. Lipoproteins differ dramatically in their density, composition, and function. They can be low and high density. So low density lipoprotein, and you can see the density, the relative density of different lipoproteins. Turns out that low density lipoproteins transport fats from the liver to tissues, while high density lipoproteins transport them back from tissues to the liver. And it turns out that higher levels of high density lipoproteins and lower levels of low density lipoproteins contribute to lower risk of cardiovascular disease. Now we're going to say a few words about steroid hormones. So steroid hormones are messengers. They're produced from cholesterol. They include various all male and female sex hormones, as well as adrenal corticosteroids, such as mineralocorticoids, such as aldosterone, and glucocorticoids, such as cortisol. So this is some structures: testosterone, male sex hormone, estradiol or estrogen; female sex hormone, progesterone, another female sex hormone; and norepinephrine, synthetic progestin. Steroid hormones can be modified into anabolic steroids. They are illegal in the United States. When you take them, they increase muscle mass. There's a bunch of side effects, such as liver damage and sleep disturbance. You can see some of them here. Okay. All these substances are banned for the general public use. Adrenal corticosteroids are produced by the adrenal glands, and these adrenal glands are located on top of each kidney. They include aldosterone. It regulates the sodium balance and water balance in the kidneys, and cortisone. It's a glucocorticoid which is produced in response to stress, and as the result increases blood glucose levels, stimulates the glycogen, not synthesis but breakdown in the liver, and stimulates the lipid breakdown, lipolysis in various tissues. So this is cortisone and this is cortisol. This is aldosterone, and this is synthetic corticosteroid prednisone that is used to treat inflammation. The last bit of today's lecture will be cell membranes. Substances can be transported across cell membrane by either diffusion or facilitated or active transport. So cell membranes separate cellular contents from the environment outside of the cell, from the external environment. Cell membranes consist of a lipid bilayer that is made of 2 rows of phospholipids. The inner portion of the cell membrane is made of the nonpolar tails of phospholipids, while the outer and inner surfaces of the cell membrane are formed by polar heads of phospholipids, and they are polar. In the fluid mosaic model of a cell membrane, there are proteins, cholesterol that are embedded into the cell membrane, with carbohydrate side chains on the surface of the cell membrane. And this lipid bilayer is actually quite heterogeneous. Proteins, carbohydrates, and cholesterol, various unsaturated fatty acids make cell membranes fluid-like. There are proteins and carbohydrates on the surface that serve as receptors for hormones and neurotransmitters, so it's actually a pretty complicated structure. The transport through the cell membrane can be passive. So this is diffusion, this so-called simple diffusion, in which particles move from a higher to lower concentration directly across the membrane. The transport can be facilitated, which is also passive, where molecules move from higher to lower concentration, this time using protein channels, which increases the rate of diffusion. Or transport can be active, in which ions move against the concentration gradient using various pumps. So this is an example of diffusion, facilitated transport, or facilitated diffusion, and active transport. Now this is the concept map. Next time we are going to talk about proteins.